Hello and welcome. In this video, I'm going to walk you through each of the questions in part one of the review for the comprehensive final exam in stats. Part one is talking about all of the non-pick the right test statistic questions. So I broke it up into two parts so the video could be a little shorter. The second part of part two of the video will be in a separate video, and that'll be on picking the right test statistic. But this one is on other types of questions. All right, so let's dive into this. Do you remember z-scores way long ago? So you need to know a little bit about those again. You need to remember that. So this question is asking if someone has a z-score of positive 0.77 on an exam, what proportion of the students who took that exam did worse, had a lower score on that exam. So quick picture for you. Draw a normal curve. Locate where positive 0.77 is. That would be about there. And the question asks for lower. So that is clearly a body. And the tail would be up on this side. So now you want to go to Appendix A. Remember that? Appendix A in the book. And find the z-score of 0.77, and then find the numbers for body under the body column. And if you do that, you will find that this is the number. Now this next question is asking about someone else, and their z-score is a negative 0.8. And this is a history exam, and that was a biology exam. It really doesn't matter, but that'll come up a little later. So negative 0.8, so let's go look at our picture again. Negative 0.8 would be on this side. And so now the body is on this side and the tail is on this side. And again, the question asks who had a lower score. So we want the tail in this case. So you're going to go look up 0.8 for the z-score. And then look up tail, and then you'll find that that is the correct number. Now, referring back to these two questions, this was Mike's score and this was Anne's score. Which one of them did better on the test relative to the other people who took their respective tests, so who had a better score? Well, the easiest way to answer that is the positive z-score is going to be better than the negative z-score. So, and scores that are more positive or further from zero are going to be better. So that's how you would answer that question. So Mike did better. After all, you could use the percentages too. He did better than about 78% of the folks, and Ann did better than, say, 21%. So clearly, that's better. All right, now we're going to jump into some jamovi stuff. All of these questions in this activity are based on a real study that was done with puppies, believe it or not. So I want to show you the study, or at least two of the tasks that were used. They're basically trying to see how good puppies are, 8.5 weeks old puppies, how good they are at following or using human social cues. Now these puppies were not trained to do what you're going to see them doing. This is like a novel experience for them. They've never done it before. And they're going to see how well they can follow a point. Do they go to what people are pointing to, or do they go to a marker? And I'll show you the two tasks. This will just take a few seconds. So here's the pointing task. So that would be a successful trial because the puppy went to the cup that the researcher was pointing at. And now in this case, they're going to do a similar task, but now they're not going to point. They're going to put a little marker or this yellow block next to the cup that they want the puppy to go to. Now, again, this is not something that they've been trained to do. this is their first time being exposed to this type of task. And so here is the marker task.
Look. Happy look. Happy look. Okay.
Okay, and those are really the two tasks that you'll sort of need to understand. There's a third task that comes up, but I don't have a video of that, so that's what we're going to have to do, just that. Okay, so now this first study or question is asking you to use a single sample t-test, and what you're going to do is you're going to be comparing the average of all the puppies' performance on the pointing task. What percentage of trials did the puppies go to the correct cup that was being pointed at? Now, you're going to be comparing that to 50% because there's only 2 cups, and so by chance you'd expect the puppy to go to the correct cup 50% of the time, and so that's what we're going to compare. We're going to compare the sample mean to that 50%, and then we're going to run it, and then you'll just plug them in down here. So let's go ahead and run it. I already have the data loaded. I did decide that I would show you how to do it just in case somebody needed a refresher, but 1 sample t, and you want to use the pointing percent variable. You want to get the mean difference, you want to get the confidence interval, you want to get the effect size, you want to get that confidence interval, you want to get all of these things. But the most important thing, you've got to remember to change this test value to the 50 that you want. If you don't, your numbers will be all wrong. So now we have all of the data that we need. And so what you need to do is fill in the blanks. So what you're going to need is the degrees of freedom, the obtained statistic, the t-value. You're going to need the p-value. Wow, that's a crazy small p-value. And there's the mean difference, confidence interval for the mean difference. There's d, and then here's the confidence interval for around d. Here is the mean. So this is the mean number of percent correct, I guess I should say, the mean percent correct for all the puppies. So about 69% of the time they went to the correct cup. And this p-value tells you that that is very unlikely to result from sampling error. A difference of being 19% higher than 50, because that's the test value, that is very unlikely to occur to sampling error. The effect size is large because it's a full standard deviation. And then you have the confidence intervals here. Even the smallest confidence interval is a large effect. So you should be very confident in the population. This is going to be a large effect. Yeah, I don't think you need anything else from that. So let's go and look. So those are all the numbers that I just pointed out, right? I don't think I explicitly mentioned the standard deviation, but it's right there. And now we have to do the interpretation. This is the harder thing. This is only worth 1 point here, but this will be worth more points on the actual test. Which of the following is the best interpretation for the p-value? Well, remember, we had a crazy small p-value. So it's going to be this one. The sample mean was higher because it was 69 than the t-test, and there was strong evidence against the null hypothesis. Really strong evidence. So yeah, that's the best one of these four. Which of the following is the best interpretation of the effect size? Well, it's a large effect size because again, it was a whole standard deviation. Again, anything close to 0.8 would be considered large. So this was definitely that. And a large effect in most cases is going to have practical importance. You might think, well, how practically important is it that we know what puppies know? Well, some people are interested in that. in the cognitive abilities of really any living organism. There's somebody studying it. Okay, next one. Based on the mean difference, p-value, and effect size, what's your conclusion? I need to take that out. That's a reminder to me that I need to add questions like this on your actual final. So that might be a clue that, yeah, you're going to have to know this. Okay, so based on all of these statistical information, what's the best answer? Well, puppies can use the social cue of pointing higher than chance. There's very strong evidence for that. Why do I add this question? Because I think people sometimes just memorize these responses and they forget to think back about, okay, so what did I learn from this? Well, this researchers learned that puppies are pretty darn good, even at 8.5 weeks, at using the social cue of pointing. That's the whole reason they did this study. Okay, so now the next set of questions is asking about an independent t-test. And again, all of this data is real. It comes from this real researchers data set. And we're going to compare the gender of the puppy, male or female puppy, on the pointing task. Are male or female puppies better at following pointing social cues? So we got to go back here and we need to analyze it. So we want to do an independent t-test now. Sex is the grouping variable. And again, the pointing task is the dependent variable. And we want all of this stuff again. And yeah, that should be fine. Now you could do a homogeneity test, but not a problem for homogeneity, right? So that means we can go ahead and keep doing the student t-test. If this were a low p-value, that means we'd have a violation of the homogeneity of variance assumption. That would mean that we have the standard deviations are very different from each other. It's clearly not the case here, but if it were the case, we might want to use a Welch's t-test instead. As you can see, they give you it's very similar answers anyway, but you never want to have both. Just pick one or the other that's the best based on this. So just to get this out of here, because we don't need it anymore, I'm going to uncheck that. And now we have everything that we need for the independent t-test question. Now, what are you going to need? Here's your degrees of freedom, and here's your t value, and here's your p value. Not a very compelling p-value, suggesting that the mean difference between performance by males and performance by females, which is down here, these are the two means, here's the mean difference, that mean difference is well within what you'd expect due to sampling error. And the again, the p-value says that because it's well within, it's not a low p-value, it's a high one, so it's probably just sampling error. Your effect size is 0.05, and again, a 0.2, around 0.2 is small, so this is a tiny effect size. And again, here are your confidence intervals. If you confidence interval around D, confidence interval around the mean difference, should you need them. All right, let's go and look at what we got. So you would enter your numbers that you need there. The evidence suggests what? Well, the data was consistent with the null hypothesis, so you definitely cannot support either of these two, because that's saying that one was better than the other. And you really don't want to say that because, again, the data was consistent with the null. Generally, when scientists talk about this being better, they mean a quote, statistically significant difference, or one that's not due to chance. So we should withhold judgment on this because the data were consistent with the null hypothesis. Again, when the data is consistent with the null, you got to realize, well, maybe I didn't have enough participants, maybe there's maybe other things. So right now, we do not have enough evidence to say that male puppies or female puppies are better on this task. So we just got to withhold judgment. We have no evidence to say that one is better than the other. So there's your independent T. Again, you should be able to interpret the effect size and the confidence intervals as well from here, but those are exactly the same types of questions that you'll see in other parts of the test where I'll ask that kind of stuff. But you should be able to do it for these as well because the interpretation is the same. Again, in this case, the confidence interval, it might be as low as a quarter of a standard deviation, that one group is better than the other, or it might be as high as a third of a standard deviation, that the other group is better than that group. So it could go either way, so you should not be very confident. All right. Now we're going to go and do a one-way ANOVA. In this one-way ANOVA, we're going to compare 3 different breeds of puppy on this pointing task. Maybe some breeds are better at using the social cue of pointing than others, and that's what we're going to ask here. So we're going to use the variable breed. There's going to be 3 different breeds. I think they were, I'll have to remember, they were a Labrador and a golden retriever, and then a mix, a golden mix, I believe. And they're doing it on the pointing task. So let's go ahead and analyze this data. So now we want to do an ANOVA. I want to go to ANOVA. Again, go to ANOVA, not one-way ANOVA. Breed is our factor. Our dependent variable, again, is still the pointing task. You want to get omega squared. You want to get all of your post hoc tests. You want to get Cohen's d and the confidence interval for Cohen's d. And you got to remember to get a no correction p-value, not a Tukey correction p-value. It's always helpful to get graphs because it helps you sort of visualize what your data is going to be like. And it's also probably a good idea for you to get the marginal mean table. That'll make things potentially a little easier for you because it makes it easier for you to find those means if you need them. All right, so here is our data. Here is the stuff you're going to have to enter into the little drop-down thing that you are used to doing for now. So here's the F. Well, here are the degrees of freedom. You remember there's 2 numbers that go in the degrees of freedom. The smaller number always goes first. So it's 2 and 161. Then you have the F value, and then you have your P, 0.005. So that's, again, a very small p-value, very compelling. And then you have an omega squared of about 5%. And again, use those guidelines to interpret that as large, medium, or small. All right, so we know now that we have very compelling evidence that, in fact, there are differences between the puppy breeds. And now as I look at the labels down here, we had a golden retriever and then a Labrador, and then the LGX, that's a mix. I believe that's a lab mix. So again, you want to interpret these. Which breed is better? Well, we'll need these details up here, but we can also come down here and look. So it looks like the golden, they have a lot of variability here, but it looks like the goldens did better than the other 2 groups. But now we got to look and see if the p-values are compelling or not. As I look at this, it looks like they're going to be, just because of the amount of overlap isn't that great here, and there's no overlap here. So I'm definitely expecting a very small p-value here, maybe a slightly higher one here, but I bet it's still going to be compelling, again, based on a very small amount of overlap between these 2 graphs, or I should say point whisker plots. All right, so this is golden and Labrador. Yes, that's compelling. So that's the difference here, golden and Labrador. That is compelling. The effect size is large. It's a 0.9. So that's a large effect. And then golden and the lab mixes, that's comparing this one to this one. Again, we know that's going to be a small p-value, and it is. And again, the goldens, so now the goldens are doing better on the task than the labs, and they're doing even, there's even a larger difference between the goldens and the lab mixes. And that is a 1.2 standard deviation. That's an even larger effect. And then you have the lab and the lab mixes, these 2. This difference, there is weak evidence for that difference. You got a 0.11 basically. That's weak evidence against the null, but it is some evidence. The effect size is small medium, so this is a good case where you'd want to definitely withhold judgment, because it might very well be the case that labs are better at this task in the population than lab mixes, and we just don't have a large enough sample. Our study isn't sensitive enough to find that difference because it's kind of small. So again, on this comparison, we should probably withhold judgment. But on these other two comparisons, we can feel pretty confident based on the p-values and the d's and the confidence interval around d that the Goldens are doing better on the task than both the labs and the lab mixes. Okay, so now let's go and see if we can answer these questions. Which of the following is the best interpretation of this one way? So it's this one down here. Golden retrievers use the pointing cues better than the lab Golden mixes, and they also did it better than the Labradors, and both differences were greater than would expected by chance, because both, or by sampling error, because both of those p-values were compelling. Okay, now again, I might ask you a question about the d next time, in addition to, or so you should be able to interpret the d, the mean differences, everything. All right, next question is going to ask about correlation. And so in this case, we are going to see if the puppy's performance in one task is correlated with their performance in another task. In other words, if a puppy does well in the pointing task, do they also tend to do well in any of the other tasks? In this case, the marker task I showed you, or there's another task I didn't show you. It's called the odor control task. And so these are all measured on % correct. And what we're doing is we're seeing if they're correlated here. Again, we're not comparing means. This is a correlation. We want to know if a given puppy does well on the pointing task, does that same puppy tend to do well on the marker task, or the order task, or the other combination between the marker task and the odor task. So we got to run a correlation here. We want to go to regression, correlation matrix. Just want to pop all of them over there. And then we get that correlation. We want to get the n, and might as well get the confidence interval as well. All right. So here we are. Now the tricky thing here is to know where to find your numbers, right? So here's the marker and the pointing task, and all the numbers you need for that. Here's the marker and odor control task, and all the numbers you need. And here's the pointing and odor control. All right, so we'll probably have to come back to these. I can't memorize all those, so we'll probably have to read what questions we need and then come back to them. So here it's just asking you for the R and the P. I could very well ask you for the confidence intervals as well. You should know where to find them. They're right here. Now, which of the above correlations is the strongest? Well, we could just look right here because these are the right answers. The strongest correlation is this correlation of 0.15, which isn't a strong correlation, but it is the strongest. All of these are positive. This 0.15 is the only one that has any kind of compelling evidence. So again, that's the one that has the strongest correlation. And again, what you're judging that on is just the absolute value of the R. If there were a negative R in here, again, you want to judge the absolute value to determine which one has the strongest correlation. Which of the following is the best interpretation of the correlation between marker and pointing? So pointing and marker right here, it's a 0.06 is the correlation, and the p-value is 0.4. That is a very high p-value. The R is very close to 0. This is very consistent with the null hypothesis, so we want to say something like that, and that's what this says. The performance of the marker increases as the performance of the marker task increases, the performance on the pointing task increases. That's just because the correlation was positive. but it wasn't a compelling p-value. The results of this trend is within that expected by sampling error. Again, because the p-value is so high. Basically, there's no real association between those two variables. They tend to be just support the null hypothesis. The data are consistent with the null. Which of the following is the best interpretation of the correlation between marker and odor control? So marker and odor control, well, I guess I should be going back here to find that. So marker, odor control, here's the correlation, here's the p-value. How do you interpret that? Well, again, this is a small correlation, small, medium, small, it's small, it's close to 0.1. And then here's your p-value, which is, it's moderately strong evidence that there does seem to be a small positive correlation between the way the puppies performed on the marker task and the odor control task, whatever that is. So we want to go down here. As performance on the marker task increases, performance on the pointing task increases. Again, they're increasing together because it was a positive correlation, but the evidence suggests is the evidence against the null is moderate. Again, because we got a 0.05 p-value. So that's the one that best fits the data. Now, now we're going to do a related samples t-test. And in this related samples t-test, we're going to see if puppies overall perform better on the pointing task or the marker task, the two tasks I showed you at the beginning. So the same puppy now is being tested on both tasks, and we want to compare the average puppy performance for each task to each other. So we can say we're asking if puppies are better at following pointing cues or marker cues. So we're going to analyze that data. We want to run a paired samples t-test. So we want to do marker task and pointing task. Those are the two we're comparing. Again, we want all these goodies. Yeah, that should be it. And so now let's look at this. We do have a compelling p-value. How exciting is that? The puppies do seem to be better at one task than the other. Which task are they better at? It looks like they're better at the marker task than the pointing task, which I find intriguing. They're better at the marking task than the pointing task. And how big is that difference? Well, that difference is about 6.8% better, and that difference of 6.8% is compelling because the p-value is crazy small. So we have very compelling evidence that they are better on the marker task than the pointing task. And the effect size is small, 0.2. Effect size is small, but because we have a pretty large sample size, we have 164 puppies in the task, we are very confident that even though this relatively small mean difference is very unlikely due to sampling error, because we have a lot of sensitivity, we have a big sample size. All right, so now let's go and look at the question here. The data suggests that puppies are better at the marker task than the pointing task, and this difference is unlikely due to sampling error because the p-value is small. And again, we know that the puppies were better on that because we could look at these means and we could look at the graph. All right, based on the statistical evidence from the related samples T you ran from the above, which of the following statements is true? The results indicate that the magnitude of the difference is about 1/4 of a standard deviation. That's a good interpretation of the effect size here. It's about 1/4 of a standard deviation. That's how big the difference was between the marker performance and the pointing performance. which the following is the best interpretation of the 95% confidence interval for these means. So again, now I'm asking you about a confidence interval. So let's go look at that confidence interval. All right, so the 95% confidence interval, there's two of them, right? You've got the mean difference confidence interval, and you've got the effect size confidence interval. So you might need to look at both of these and then choose the interpretation that I've written up that best matches one of them, right? So if you look at the D one, it might be a small difference of about 0.1 of a standard deviation in the population, or it might be a medium difference of about a 0.4 standard deviation, the difference between their performance on these two tasks in the population. This confidence interval means that the difference in the population might be a difference of about 3%. And in this one, it might be a difference of about 11%. Okay, this is the upper bound and this is the lower bound. Now you got to be careful here. This mean difference, because the dependent variable is measured in percent correct, these mean difference confidence intervals are percentages. The mean difference for D these are not percentages. These are standard deviation units. So you got to read the answers you're going to get next carefully so that you aren't fooled by that. All right, let's get out of that. All right, so which of the following is the best interpretation for 95% confidence interval? This one's going to be the best, but let's talk about these others because I'm going to alert you to some things. The mean difference in the population could plausibly be a mean difference of nearly 42%. That's not true. It's not the mean difference. The mean difference, there's the mean difference. That's not 42%. That's a FOIL, right? That's a 42, but that's not even a 42%. That's half of that's a 0.4 of a standard deviation. So that's just completely wrong. The mean difference is probably just created by sampling error. That's not something that a confidence interval tells you. That's something you look at for a p-value. The mean difference of this size would occur extremely rarely if the null hypothesis were true and there were no methodological flaws. This is also something that a p-value tells you, not a confidence interval. So by eliminating these, you know this is right, but this is maybe a different interpretation of a confidence interval that you've seen. Normally I say it's the plausible value, it's a range of plausible values, which is true, but here is another good one. The best guess for the mean difference in the population is this. Now, why do I say that? Well, the mean difference is this, right? That's the mean difference. Here's the confidence interval around the mean difference. The confidence interval, the mean difference here is the point estimate of the confidence interval. It is the middle of these two values exactly. And that means that it is your best guess. That is your best guess at the population mean difference. It might be as low as this. It might be as high as this, but this is your best guess. Okay. Now we are ready to do a two-way Anova. And in this one, our two independent variables are breed, two different types of breeds. And I have to confess, this is made up data. All of the data we've done so far was actual data from their study, but they didn't have the right data for me to do a two-way Anova, so I just made it up. But I'm telling you now, so that you don't mistakenly tell people about these results. All the other results you can tell somebody about if you care. All right, so we're going to have one independent variable, which is breed. We're going to have two different breeds. And then we're going to have these two different breeds perform two different cognitive tasks. And then we're going to measure their performance on the different tasks. And we're going to see is one breed better than another breed on one task or the other? Do breed and cognitive task interact to influence percent correct on a given task? So we need to run this test, of course. Now we need to get a new data set. So I had to make this data set. And I used to have a greyhound. So I chose that as one of the breeds. And then another one is the poodle. And then there's two different tasks. They're performed, and you have different dogs in each task. So you have a greyhound performing a bunch of greyhounds performing the pointing task, a bunch of poodles performing the pointing task, a bunch of greyhounds performing the marker task, and then a bunch of greyhounds performing and then a bunch of poodles. You got the 2x2. Okay, so now we want to analyze it, of course. So we wanted to go one way again. The two factors are the breed and the task, and the performance on those whatever task they did is the dependent variable. We want omega squared. We want to get all of the post hoc tests. We do not want correction p-value, and we want the d and the confidence interval around d. And you definitely want to get a graph here. I think that'll help you interpret the interaction if there is one. Okay. And so here we are. Remember, you're going to get 3 f values. One that is talking about the interaction between breed and task. And you can see here we have a p-value. So we're going to have a significant interaction. We're going to have an interaction that is going to have compelling evidence. And then we're going to have a main effect of task that's not compelling. And we're going to have a main effect of breed that is compelling. So one of the breeds overall did better than the others. Only weak evidence for a difference in task in this data set. And then we do have compelling evidence for an interaction. Let's go ahead and go down and look at that interaction, because that's probably the question I asked first, if I had to guess. It looks like, here's the interaction. Looks like greyhounds did about equally well on the marker task and the pointing task. However, poodles seem to do a lot better on the marker task than the pointing task. So that's probably the interaction again. We're going to have to go up and look at the p-values to see for greyhounds, is this difference compelling? Probably not. Look at the overlap. So this is probably going to be a large p-value, and here hardly any overlap, so this is probably going to be a small p-value. So probably what we're going to find when we go up and look at the p-values is that for greyhounds, they did about equally well. They maybe did a little better, but the difference between the tasks are just due to sampling error. They did a little bit better in the pointing task, but the difference is probably just sampling error, I should say. However, the poodles did a lot better, it looks like, for the marker task than the pointing task. But again, you should go up and look at the simple effect p-values, and you're going to find those right here. So greyhound marker, greyhound pointing. Here's your p-value, 0.5, not compelling at all. Here is your D, not compelling, very small effect. So that's about what we expected. And now what about the other simple effect? Poodles and marker task, poodle and pointing task. The mean difference is 8%. That is compelling, and it's a medium effect size. So now that's pretty much the interpretation of the effect size, right? Okay, so you got to, I never pointed that out. Shame on me. Okay, so, oops, wrong data file. So where do you find all those numbers? Well, for the interaction, it's going to be 1 and 256. Here's your F value, which is next. Here's your P value. Here's your mean square error, mean square residual, your omega squared. That's everything. All right, now, which of the following is the best interpretation of the interaction? It's this first one. Greyhounds, the performance difference between the marker task was within that expected by sampling error, and this difference has a small effect. However, poodles performed much better in the marker task than the pointing task, and this difference was bigger than expected by sampling error, and this effect size was medium. It's exactly what we just said. These others are, this is an incorrect interpretation of the interaction, and then these others are really all main effect interpretations, and then one outcome that just says the data is consistent with the null. Remember, you have 3 statistical tests, and each test has its own null hypothesis. Okay, so next question. Which of the following is the best interpretation of the main effect of breed? Okay, so let's go look at that. Main effect of breed. This one's the compelling one. So you might want, and again, we want to go down and look for the main effect of breed. So it looks like overall, poodles are doing better on the tasks. Overall, on both tasks, we're collapsing across tasks. Poodles did better than greyhounds. And what's the effect size of that? Well, it's about that, 0.04. So you should use your cut lines for that to determine whether that's large, medium, or small. All right, so overall, poodles did better on cognitive tasks. This difference was more than expected by sampling error with a medium effect size. Okay. Which of the following is the best interpretation for the main effect of cognitive task? Well, let's go up and look at that. Okay, cognitive task, 1 and 256. Your F. Not compelling. This is weak evidence at best. You know, it's at best, it's very weak. It's got to be close to 0.1, and we say that if something's close to 0.2, we say it's consistent with the null. So this is very weak, if anything. And if you look at the task, if anything, they did better on the marker than the pointing. But again, that p-value, you should probably withhold your judgment on this because the p-value is not compelling. Maybe they would do better if we had a bigger sample and we had enough statistical sensitivity to detect this difference. But it looks like this difference on task is very small. So that might not have practical importance, something that's that small. So let's go back and see what we did here. Yeah, it looks like best interpretation of the cognitive task, the data are consistent with the null. That's the best answer here. Okay, and now the rest of the questions are just general questions about how to interpret p-values and basically the pillars and just generic questions. It's not based on a data set per se. They're just based on general knowledge I hope you have learned about various statistical concepts. This one is which of the following is the most accurate description of a p-value, and what does the p-value tell you? Well, I'll just walk through each of these quick. They describe the practical importance. No, that's what an effect size does. They estimate the values of the population. No, that's what a confidence interval does. They reveal if a study is likely to have methodological flaws. No statistic does that. They indicate the probability of a given outcome, assuming the null hypothesis is true, and there's no methodological flaws. That's true. That's basically the definition of a p-value. And they suggest that a study's results match those of a previous study. No, you need the results, but you need to compare the results to the previous study. And it's not a very good idea to compare p-values. You should compare effect sizes, not p-values, to decide things like that. Which of the following is the most accurate description of confidence interval? Well, I said that. It's this one. Which of the following is the accurate description of an effect size the researcher tells you? Well, this one. I said that as well. And again, you should be able to do more than just memorize these. You've got to understand what they mean. I might word it differently, but it's going to mean the same thing. So don't just memorize the words. Understand what it means. Ooh, central limit theorem. That's a blast from your past, right? What's the central limit theorem describe? what does it describe of the following? Well, it does all of them. Again, remember what the central limit theorem is. It's really important because it tells you that a distribution of sample means is going to have a normal shape. And it also tells you the specific spread of the distribution of sample means, which is the standard error of the mean, standard deviation divided by the square root of n for like a single sample t. And then it also tells you the center of the distribution of sample means, the mean of the, it's all of these things can be derived from the central limit theorem. So that's the correct answer there. And our final question, which of the following is the accurate description of the distribution of sample means? Well, it is all of the sample means that are possible with the sample size you're using. All right, so boy, I can't believe I went on for 45 minutes. If you're still here, you are a trooper. I will also be releasing a second video because I know you want more of when I go over the choose the right test statistic questions, which I will post alongside this one. All right, again, I won't be in class to answer questions this next week, but I will be online during class period the first several minutes, and I'll stay as long as people are there to ask questions if they want them. But again, I'm pulled away on interviewing duty, so I'll be there. All right. Thanks. I hope things go well. Email me if you have problems or show up at class time. Thanks.